got dinner is mission evening everyone, and welcome to our second VSNI webinar. Tonight we will talk about some companion animal parasiticides, their presence in the environment, and some strategies to mitigate this risk. So my name is Niamh. I am a vet and co-founder of the Veterinary Sustainability Network in Ireland, and I will be chairing tonight's session. Thank you all for joining us this evening to learn about this increasingly important subject. A special thank you to our three speakers, Rose, Jason and Sarah, to Excel Vets and our webinar committee for helping organise this event, and also to Diagnostic, who have supported the running of this event. So just a quick run through our agenda. So we'll start with just a little bit of housekeeping first. I'll give you a very, very brief introduction to the Veterinary Sustainability Network and a very brief introduction into the European Medicine Agency review into companion animal parasiticides. And then we'll move on to the important part of our evening, so our three speakers. At the end, we will have a time for a Q&A. So if you'd like to add some comments, or questions into the question box throughout the webinar, please do feel free to add some things in there. So just some housekeeping. So this session is being recorded. You're all muted on entry to reduce background noise, and please keep your mics on mute for a smooth experience for everyone. This session will last about 50 minutes, with time for Q&A there afterwards at the end. As I said, use the chat box to say hi, ask questions, make comments during the session, and we'll answer hopefully all the questions, as many as we can get to during the Q&A. Please stay respectful to ensure a positive learning experience for everyone. If you have any technical issues, message us in the chat, and our backroom team will help you out. If you get disconnected, just rejoin using the original link. We'll send out a recording, the slides, and the resource links a few days after the session as well. So just a little introduction to the Veterinary Sustainability Network, for those of you that might be new here or mightn't have heard of us as of yet. So the Veterinary Sustainability Network Ireland is a not-for-profit organisation. We were founded in 2024. Our mission is to be a leading voice for nature and the environment in the veterinary industry in Ireland. We aim to create a veterinary sustainability network by engaging and educating those in the sector and collaborating with stakeholders inside and outside the veterinary field on this topic. And just to give you a quick background into this talk, companion animals, parasiticide and biodiversity. So to put it into context, the European Medicines Agency is currently holding a public consultation on changing the investigations into companion animal parasiticides. So basically, all veterinary medicines applying for a license must undergo an environmental assessment. All of these are subject to phase 1 assessments, which is, i.e., will the use of this product result in any potential environmental exposure or contamination? If the answer is yes, they move on to phase 2 assessments, which is if environmental exposure is expected, could the product pose a risk to the environment or non-target species? Interestingly, all companion animal products currently stop at phase 1 assessment. And as this is stated in the guideline, that because companion animals are not intensively reared and treatment is usually individual treatments, they are unlikely to result in environmental harm. However, we are finding that some of these products from the companion animal sector are possibly being found in concerning concentrations in the environment, which will be elaborated on by Dr. Rosemary Perkins this evening. Therefore, in light of the emerging research on this topic, the European Medicines Agency are looking to change these guidelines in relation to companion animal parasiticides and are currently holding a public consultation on the proposed methodology. So to increase awareness on this issue, we've decided to run this webinar on this topic. So that's it for me for the moment. And I'm delighted to hand you over to our speakers who will share their expertise and insights in this area. Firstly, we'll have Dr. Rosemary Perkins, then Dr. Jason Keegan, and then Dr. Sarah O'Dwyer. Just to introduce Rose to you all. So Rose is a veterinary surgeon and Research Associate at the University of Sussex. She's earned her PhD studying environmental emissions from pet parasiticides and has authored numerous publications on the topic, including research on waterway pollution from pet parasiticides through down the drain and swimming pathways. She is an active member of the Imperial College PrEP group and Vet Sustain's Pet Parasiticides Working Group. Over to you, Rose. Thank you.
Thanks, Neve. Share my screen. Yeah, so good evening, everyone, and thank you very much for inviting me to speak. My name is Rose Perkins. I'm a vet and a research associate at the University of Sussex. So today I'll be looking at pet parasiticides from a One Health perspective. So this puts their use in a broader context, which considers human, animal, and environmental health. So parasiticides kill parasites. They are very useful drugs. They do have a place, but we want to use them in a way that is sustainable and responsible, that minimizes unintended harm, such as environmental contamination, and also ensures that we maintain their efficacy so that we have effective parasiticides when we really need them. So in this talk, I'll mainly be looking at environmental emissions for topical pet parasiticides, which is my research area. I'll also be looking a bit further at other potential effects or impacts associated with parasiticides, so putting it all in the One Health context. And I'll also be flagging some of the knowledge gaps around this. And lastly, I'll have a brief look at what a One Health risk-based approach to parasite control in pets may involve, and how this ties in with the concept of contextualized care. So we know that medicines and the environment are quite a serious emerging concern.
Sorry, Rose.
Chemicals.
I'm just going to stop you there. I don't think your slides are up, Rose. Can anyone?
Oh, I'm so sorry.
See the slides?
I thought I'd shared those.
No.
There we go.
Perfect.
Can you see that? There we go. My apologies. So medicines are specifically designed to have biological effects, and if they pass into the environment, they can cause harm. This diagram illustrates some of the pathways through which medicines can enter the environment. For example, this shows the case of diclofenac used in cattle in India, which caused the collapse of vulture populations in the 1990s after exposure through contaminated livestock carcasses. And human pharmaceuticals often end up going down sewers and into wastewater, where in many cases they are not removed through wastewater treatment. For example, steroidal estrogens used in contraception pills have been shown to cause feminization of male fish, potentially affecting fish populations. And what's quite striking in this diagram, which is from 2015, is the fact that it mentions medicines used in livestock and in humans, but there's no mention of medicines used in pets, which is curious, as we use a lot of medicines in pets. There are probably over 1 billion pets on the planet. So the reason for this does warrant a bit of scrutiny. So as Neve touched on, this may have something to do with their regulatory framework. So before veterinary medicines are approved for use, they are required to undergo an environmental risk assessment. This assessment assumes that medicines intended for use in companion animals are unlikely to enter the environment in significant quantities, so negligible exposure is assumed. And therefore, even in the case of potentially hazardous compounds such as parasiticides, the risk to the environment has generally been considered too low to be of concern, so no information is typically required on the ecotoxicity or potential routes to the environment. And this international framework is being reviewed, but it is still in place, so we still don't know much about the risks posed by most companion animal medicines. So that brings me to my research. This focused on two particular veterinary medicines. Fipronil and imidacloprid are parasiticides widely used in spot-on flea and tick treatments. They were originally developed for use in crops as agricultural insecticides and were very widely used for that purpose. And then following evidence of environmental harm resulting from their agricultural use, particularly impacts on pollinators, eventually a total ban on agricultural use was introduced in the EU and the UK by 2018. However, following agricultural restrictions, something strange was found. Because of their historical agricultural use, they were still monitored for in the environment, and high levels of fipronil and imidacloprid were found in UK waterways after the ban, and frequently occurring at levels exceeding recognised safe environmental thresholds. And particularly high levels were occurring in urban areas, which was also unexpected for pesticides that had primarily been associated with agriculture. And this report from the UK water industry found high levels of fipronil and imidacloprid in wastewater effluent, so in treated sewage, and downstream of sewage treatment works across the country, demonstrating that they're getting into rivers through wastewater and very little is removed through the treatment process. So the question was, how are they getting into wastewater? So this diagram maps out some of the potential pathways from spot-on treated pets to the environment. As you can see, it's a complex situation and there are multiple potential routes to waterways. After application, they spread over the coat and skin and disseminate into the surroundings, including the pet's bedding and owner's hands, and ultimately some washes down the drain and into wastewater. And because they are extremely potent insecticides, even just a portion of the applied amount passing down the drain can be significant, especially if many pets are being treated. So my research examined and quantified some of these down the drain pathways. So we measured wash off through bathing of dogs, washing of dogs' bedding, and through hand washing before and after petting their dogs. And these graphs show the percentage of the applied amount that washes off over time for these different pathways. As you can see, the wash off decreases over time for most pathways, but occurred through all of these pathways for at least 28 days after spot-on application. And by modelling the data, we were able to show that these pathways are a major source of measured wastewater pollution. We also investigated swimming emissions and found that environmentally significant quantities of fipronil and imidacloprid can wash off through swimming for at least 4 weeks after spot-on application, which indicates that the product usage guidelines, which usually allow swimming for around 2 days after application, don't provide adequate environmental protection. And this was supported by a study at Hampstead Heath, which found really high levels of both of these insecticides in dog swimming ponds and nothing in non-dog swimming ponds. So what are the effects of these chemicals on aquatic communities? We don't actually have any research looking at their specific impacts in UK waterways yet, but we know from research elsewhere in the world that impacts are likely to be occurring with the levels that we're finding. Large-scale studies from Europe have indicated a significant negative correlation between imidacloprid concentrations in surface water and the abundance of aquatic macroinvertebrates, and with declines in insectivorous bird populations, with these effects being found at concentrations below those regularly occurring in UK waterways. Similarly, studies have also found that even very low concentrations of fipronil degrade stream communities in multiple regions of the US, again at concentrations below those found in the UK. So there's good reason to believe that the measured levels of these chemicals are likely to be having an impact. And comparative environmental risk rankings have identified both of these chemicals as being of high priority compared to other pollutants. So, for example, Dave Spurgeon performed an environmental risk ranking of all the organic compounds in the Environment Agency LCMS monitoring dataset for England. This dataset included over 600 chemicals of concern, including pesticides used on crops and human pharmaceuticals, amongst others, and found that fipronil was the organic chemical posing the greatest threat to surface water ecosystems, based on how frequently it was being detected and the concentrations that were found. Another study looking at emerging contaminants of concern in London's waterways placed imidacloprid at the top of that ranking. But water pollution isn't the only concern. The study found a high prevalence of pesticides used in veterinary medicines, including fipronil and imidacloprid in hair-lining birds' nests, and found potential associations with increased tick mortality. So we are still very much in the early days of this research. Because of the historic assumption of negligible environmental risk from these products, very little research has been done on their impact. We have very little data and there are many knowledge gaps. So putting this into a One Health context, this diagram lays out risks and benefits of parasiticides. We can see that it covers all 3 aspects of One Health. So benefits are reduced infestations, potentially reduced zoonotic disease. Risks include terrestrial and aquatic contamination. So soil pollution is a big unknown. Many parasiticides, including wormers, are excreted in feces and may contaminate soil. We know that wormers such as praziquantel and pyrantel are toxic to earthworms, and macrocyclic lactones are toxic to dung beetles. These are all extensively used in small animals, but there's currently no research on their presence in soil or the impacts they may be having. Resistance is another concern. So this hasn't received much attention in small animals, but there is evidence that this is an emerging concern. So tick resistance to fipronil has been reported in several countries recently, including the US and Brazil. Anthelmintic resistance in hookworm affecting dogs is now widespread in the US and Australia, and that has become quite a serious concern. Resistance in Dipallidium caninum tapeworm and heartworm has been reported in the US and recently in Europe. So this is certainly not something that we should be complacent about. Research on resistance is very limited in the UK, but one study showed widespread treatment failure for fipronil and flea infestations, where over 60% of cats and 45% of dogs treated with fipronil still had fleas. However, no research has investigated whether this is due to resistance or to operator error, And so this is another area very much in need of research. And then finally, there's a question of human exposure to parasiticides. So these products are not designed to harm humans and have generally been considered low risk. However, chronic pesticide exposure has been implicated in a range of human health effects. And we know that topically applied parasiticides can disseminate widely in the homes of treated pets, and that families can be exposed for long periods of time. And so there is a risk of chronic low-level exposure. One area of particular concern is prenatal and early life exposure to pesticides, especially the potential for neurodevelopmental impacts. And a few studies have looked specifically at flea and tick treatments and found potential associations between maternal exposure and neurodevelopmental effects. So this is another area that really does need more research. Finally, I want to touch on risk-based parasite control. So this is the approach advised by veterinary associations such as BBA and BSABA, and is now required by the UK's RCVS practice standard scheme. It means moving away from blanket treatments, like treating all animals all year round, and targeting preventative use towards higher risk animals. Effectively, it's a compromise between the old approach of blanket treatment and its opposite, which is treatment based solely on diagnosis. There is a lot of discussion to be had on where that balance lies and how much risk we should tolerate and what risks are acceptable. This is an aspect of contextualized care, and that vets should be discussing the needs of individual pets with clients and including them in that decision-making process, taking into account their concerns and their circumstances. Pet owners should be informed about risks and benefits of parasiticides to support informed choices. So to summarize, this is a very complex and challenging One Health area, but awareness of the challenges is certainly growing. We've seen through the progress that has been made with veterinary antibiotic use that it is possible to develop more sustainable approaches in light of new information. So thank you very much for listening.
Thank you very much, Rose. That was really informative. Thank you. And it's great to see the research that's been done in this area, and hopefully we see some changes soon in the amount of the concentration in the waterways. So next up we have Dr. Jason Keegan. So Jason is a zoologist and postdoctoral researcher. His research interests include the epidemiology of parasites of veterinary and medical importance. His work has been published in various international journals, including Veterinary Parasitology and the Journal of Helminthology. He is currently investigating the level of soil contamination with the eggs of Toxocara in the parks of Dublin. Do you want to pop up your slides, Jason?
And you see them there?
Yep, all good.
All good. All right, well, good evening, everyone, and thanks to the VSNI for inviting me to give this talk. So I'll be talking a bit about my research history and how it sort of impacts the One Health perspective on companion animal parasite control. So my sort of first outing into parasitology, or into companion animal parasitology at least, was during my master's. And for this, we were looking for Toxocara eggs in the hair of owned dogs. So previous work before me had found lots of eggs, lots of infective eggs in stray dog hair. So we wanted to extend that to owned dogs to see if a similar level of risk was present. The good news is the similar level of risk was not present. Out of about 184 dogs, we found 8% had eggs, but none of these eggs were none of them were infective at the stage that we found them. So there was a variety of kind of hair studies going on at the time, and basically they all kind of generally came to the conclusion it's not a major route of transmission. So then eventually I went back to soil, which is probably the most, one of the most significant routes of transmission. Just in between that, I've worked on some projects involving looking for the prevalence of Echinococcus in Ireland and fox populations to ensure that we are still free of it, so that we maintain our derogation so that animals get wormed to prevent us importing this parasite that can cause quite serious human illness. And then I moved away from the companion animals for a while and did my PhD on anthelmintic resistance in sheep nematodes in Ireland. And we showed that there was a huge amount of treatment failure on sheep farms, as well as identifying some of the first examples, or their first example of multi-drug resistance in sheep nematodes in Ireland. So, the way we use these drugs is important. We need to learn these lessons, and we should start extending them to companion animal parasites as well. So back to the work that I'm on at the moment is my postdoctoral research, and the aim of this was to look at the degree of environmental contamination with the eggs of Toxocara in Dublin city parks. So Toxocara infections. The Toxocara, it can infect dogs. It's their definitive host. They reproduce and produce eggs from dogs. They can also infect lots of other mammals, warm-blooded mammals. But within any other animal, these parasites do not complete their life cycle. So what happens is the larvae start to migrate through their body. They don't know where to go, and they cause illness. So adult dogs normally don't get too badly affected. Puppies do. Same with cats. But when humans get infected, again, most of us actually won't have any symptoms, but some of us will. And there's a number of different syndromes that are associated. So ocular larval migrans is one of the main ones, and Toxocara, the old wives' tale, is if you wash your hands and if you touch a dog and don't wash your hands, you could go blind. And it's rare, but about 70 cases of blindness do happen in the US as a result of Toxocara. As I said, it's probably not from touching them based on my previous research, but you would probably get these things from other places. But there's other types of disease that humans can get. So visceral larval migrans, if the larvae are migrating through other organs in the body, causing damage there. And then covert toxoplasmosis is quite hard to diagnose, hence the name. These can go really undiagnosed, they're underreported. It's really difficult to know, but like there is also evidence that it can impact your cognitive development in children. So it is quite an important disease that we know very little about, especially considering how common it is, which I'll get into a bit later. So the ways we get infected, we think the primary routes of transmission is direct contact with soil. So these eggs in the environment that are infective, we accidentally ingest them. Also through the consumption of unwashed vegetables. So there's more data coming out these days about food from gardens, food from agricultural, large farms, and we're finding eggs of Toxocara on them. And even some bags that are washed and ready to eat. So contact or eating vegetables, fresh vegetables, is possibly, or unwashed fresh vegetables anyway, is a possible route of transmission too. Eggs and dog hair, as I mentioned, could possibly cause it, but it doesn't seem to be a major route. Then there's also the ingestion of larvae from parotenic hosts. So the Toxocara larvae can live in various farm animals, and if you don't cook that meat properly and there's a larva present, you can become infected via that route too. And the same way with the dogs. If the dogs eat that meat, they can become infected. So that's why the idea of them eating rare meat is important. This is a map of the sero prevalence of Toxocara infection in humans around the world. And in general, about a fifth of the world's population is seropositive to it. Now, again, most of these people probably don't have symptoms, but again, it's understudied, so we don't really know that well. As usual with these parasites, these soil transmitted parasites, within the tropics, the environment is warm and humid. So it can encourage their development and growth in the environment and their maintenance. But also in Ireland, because of our mild climate, they can persist quite longer in the environment as well without dying. So the environmental contamination can persist for quite a while. But in terms of sheer numbers, based on these seroprevalence estimates, we've hundreds of millions of people worldwide are or have been infected with Toxocara, especially even including industrialized countries. So rivaling malaria in terms of absolute numbers. Now, obviously, malaria is considerably more pathogenic, but in terms of numbers, this illness is likely to have an effect. So during this research, while I was starting out, one of the undergrad students, was during COVID. So your choice of final year project was limited. So they did a survey of dog owners about their parasite control practices. So this was done in January 2022, and in that 1 month, we got 662 responses. I'm trying to do surveys now, and I can't get anywhere near that in more than 1 month. So that was a good time to be doing surveys. But of these respondents, about 52% of these Irish respondents said that they worm between 0 and twice a year. So that amount of worming is unlikely to reduce egg shedding in the environment. And additionally then, the majority of dog owners did report disposing of their feces correctly, of the dog's feces correctly, so at 71%. Now this is self-reported data as well, so we don't know how many of them actually do it. But that leaves about 30% that didn't. And then one of the interesting findings of the study was 33% of people from rural environments said that they never picked up after their dog at all, in comparison to 3% of people in urban locations. So I mean, there's varied risk. There's probably more risk of people in urban, the higher concentration of dogs in urban locations. So it's more important that we pick up after ourselves when we're in urban environments, because the chance of infection is higher. But if your dog is roaming a forest where someone rarely goes, then the chance is slim. But moving on from the dogs, then I started looking in the soil. So these are just other things. These are not parasites, but this is just a variety of life that you can see in normal soil in a Dublin city park. I wasn't expecting it to be so like vibrant and diverse. Lots of different mites, some caterpillars looking back at you, and this, what I can only assume is some sort of alien species. But what I was looking for was this, and this is the eggs of Toxicara, and these are Toxicara canis eggs. Um, and you can see a couple of different versions here. So this is an unembryonated Toxicara egg on the top right. One cell inside. It's not currently infective. So when these eggs are released in the feces, they need a period of time under appropriate environmental conditions, so temperature and humidity, before they can start to develop. So here you can see the cell is starting to divide, and here you can see a number of cells within the egg shell. The characteristic kind of crystal golf ball shell is what it's sort of known as. But if you look closely here, you can see the motile larva inside the egg, moving around, waiting to be ingested by a dog or a human to go on and cause infection. There's also a couple of other eggs in and around. So many different eggs in these soil samples. It was just bizarre to see how these samples were teeming with life, even in the middle of the city. So we looked at a number of different parks, and we looked at certain locations within those parks to see if areas were higher, more likely to have infection or to have contamination with Toxicara eggs. And what we found was the entrances were the most heavily contaminated. And we kind of put this down. A lot of anecdotal reports from dog owners will say they'll bring their dog for a walk, but the dog will wait to go to the toilet until it gets into the park. And this is kind of borne out by the results, because around the entrances we found most of the eggs. So of the 12 parks we studied in detail, 9 of them were positive for Toxicara. It was about 5% of all the samples. I also collected fecal samples from wherever we were collecting the soil samples from, and that resulted in 46 fecal samples collected altogether, only one of which was positive for Toxicara. Um, so then overall, we got 92 eggs, and 90.2% of those 92 eggs were found to be potentially infective. So they had motile larvae in them. So I think the highest number we found by one entrance is maybe 15, but it's possible that one egg can actually go on and cause disease. So it's still important to try and reduce this level of environmental contamination. So we published this recently enough, and that's that there. But the most important thing that this is being caused by dog fouling, and it's a really kind of interesting thing. Like it's one of the most commonly complained about things in most county councils. Everybody hates it. Like nobody is happy about the degree of dog fouling that is present. And as I mentioned earlier, still some people report not picking up after their dogs, despite everyone hating dog poo being everywhere. But despite this, the prevalence of dog poo and hatred towards it, there's been very few good studies. So in 2011, there was this systematic review of studies to see how you can reduce dog fouling in public, and none of the studies were good enough to be included in the systematic review. Now, since then, there were 68 studies that were looked into, and none of them were good enough. But there have been a couple of other sort of studies since then. This is the biggest one, and of interventions that can decrease dog fouling. This is something called the Poo Patrol. It was run in Bristol, and it involved sort of community organisation involving schools, educating them about the parasites, educating them about science, about going out and counting things. So they got them to do art, they got them to make posters about dog poo and encouraging people to pick up after their dogs. And these were all put around the locality. And then they also counted any instance of poo they saw. They also sprayed it with colourful, environmentally friendly spray paint. So they did that, counted them all, and then went back 2 weeks later and did it again. And they found in most of the locations there was a 65% reduction in the amount of dog fouling. So a huge impact of this kind of work. And this is one of the only studies that has been done on this. There are a couple of other options as well. So this is a smaller study. It was done in 17 locations, and it was simply the use of these posters that have eyes on them, and then messages underneath, like telling people that someone is watching. So this idea of people being watched can make them change their behavior. And these are found to be effective. They found to reduce dog fouling by about 46%. And the most effective poster was the one with some sort of positive or affirming messages with telling this is what other people do, why aren't you doing it? And then the only other study that I've been able to find is this Slovakian study where they got bright, glowing footprints that would go towards the closest bin that you could put dog poo in, and that sort of facilitated people finding where to find the nearest bin. So just making it easy for people to clean up after their dogs is probably the first thing we can do to help reduce environmental contamination with the eggs of Toxocara, but also whatever else happens to be in the dog feces, like parasiticides. Just another example. This is a, a way of decreasing, an intervention that can decrease dog fouling. It's a lamp in a park in Malvern in the UK. And if you get your dog poo, you have a little brown bag that is provided with it. You can put your dog poo in the lamp and it powers the lamp. It's a gas-powered lamp from dog poo. So we could, introduce these in certain places and see if that had an effect too. But just to introduce the sort of idea of over-distribution of parasites in hosts, this is a common occurrence or a common trend in lots of different helminth or species, for example, anyway. You'll find the majority of hosts won't have any parasites whatsoever, but then you'll find a small number of hosts will have huge numbers of parasites. So what we want to try and do is identify these ones down here at this end of the graph to see which ones are which ones are responsible for most of the environmental contamination, and then we can treat them accordingly, according to their own individual biology. And this is the most recent guidelines from ESCAP. So I'm also on the ESCAP committee of the ESCAP UK and Ireland committee. And this is the 2025 guidelines for all of Europe. They might be, because we've some different parasites in here, as I mentioned, we don't have a kind of Coccus multilocularis here yet. So there are some things in Ireland that would be less risky than in mainland Europe, for example. But again, this kind of recommends an individualized risk-based approach. So a conversation between the vet and the owner about the circumstances of your ownership, of the risks to the owner and the risks to the animal, and based on the animal's behavior. So whether they eat raw meat, whether they go outside, and things like that. So and the riskiest group that are roaming outside and eating raw meat all the time and exposed to tapeworms, the only way you can be sure that they won't release eggs is to treat them every month. So that's quite a lot of treatment that you might want to avoid. So in all of these circumstances, they're also recommending that you could also test and treat, and encouraging kind of more data collection before, instead of just blanket treating just because we've always done it. So more information is definitely a good thing. So these guidelines are available for dogs and cats, but it leads us to there are still sort of a number of knowledge gaps that we need to address. So even in Ireland specifically, the most recent studies of intestinal helminth prevalences are done on a mixture of pet and stray dogs. So we don't really know the picture in pet dogs anytime recently. Also for adult dogs, we don't really know the egg shedding patterns. Likely some of them might shed eggs for a long time, but a lot of them probably don't. So we might be giving them needlessly giving them wormers if they're not going to ever shed eggs. Again, anthelminthic efficacy is not something that we test a lot for in dogs, so this is kind of something that we're moving to encourage people to do more. So the idea now is to kind of get more data. So more real-world fecal egg count data would really help us inform treatment protocols that can reduce the risk of infection to dogs and human, but also reduce the amount of parasiticides getting into the environment. And on that note, I'll lead into Sarah, who's going to talk to you about some counting techniques. Thank you very much. Oh, sorry, just finally to mention, if we're also trying to do a survey, as I mentioned earlier on, it's very difficult to get people to do survey these days. So we need vets to respond to this. There's a QR code there. It'll go out, you'll get it in in the slides afterwards. There's also a link to the ESCAP webpage there, and there are certain grants available for summer students, and there's awards for parasite control programs in veterinary practices. So if you've introduced a new parasite control program, you can submit your program to ESCAP and you might win a £500 prize. So that'll be awarded every year. So keep an eye on that. There are potential benefits available to you all, and I'll leave it there. Thanks a million.
Thank you very much, Jason. I really love the poo patrol bit as well. It really shows the kind of the multimodal approach that we can take, that we don't always have to rely on medicines. I think it's fantastic. And it also shows the power of children as well.
Exactly.
So I'm going to hand over to our final speaker of the night, who is Sarah O'Dwyer, who is joining us, I believe, from Australia. So thanks a million for that. Sarah is a vet who's worked both in equine and companion animal practice. She has a certificate in advanced veterinary practice in equine internal medicine and is a member of the Australian and New Zealand College of Veterinary Scientists. Sarah currently works as a veterinary technical advisor at Diagnostic Limited, who are kindly supporting this webinar this evening. So I'll hand over to you, Sarah.
Okay, great. Is my screen okay?
All good from me, yeah.
Fab. Okay, so the title of my talk is Diagnostic Options for Assessing Parasite Burdens in Companion Animals, and I've renamed it What's in My Pooch's Poo and My Moggy's Boggy. So I think just, first of all, I'm speaking as a first opinion small animal vet and an equine vet. So for me, the worms of veterinary significance in dogs and cats in Ireland at the moment, and we could talk about everything, Giardia and Isospora, but really it's roundworms, including Toxocara species, your hookworms, your whipworms, and then your tapeworm, Diphylidium caninum, and then your lungworm. But in this talk, I'm just going to talk about roundworms because I think it'll just be too broad, and there'll also be a survey. So if you could have your phones ready, it's just a short survey, which I think will make it a little bit more interesting. So at the moment, what is the roundworm control in companion animals in Ireland? So at the moment, it's preventative treatment, and that is the backbone of the control. So it's a routine treatment, which comes from either habit or culture, or that expectation from owners, or maybe worry that if you're not treating, there may be reprisals. And the anthelmintic treatment schemes that we use are often designed to reduce zoonotic potential of these intestinal parasites, as well as to improve pet health welfare. So it's very rare that we see, overt clinical disease caused by parasites in the most of our populations of dogs and cats, unless, they're young puppies and kittens, or they're strays, or they're in kennels. And at the moment, I feel, in my experience, having moved from equine to small animal, that fecal testing as part of this parasitic control program and diagnostic workup is overlooked compared to large animals. And also that individual risk assessment is minimal when we're dispensing out parasiticides to healthy animals. So this is the first survey, so let's see if it works. And it's, how often do you perform fecal testing for parasite screening in dogs and cats before prescribing anthelmintics? So I can leave up the QR code there for a second or two, or you can join, and I think Jason's going to put the link in the chat.
Yeah, so there's a link to the chat. If you go to a browser and just type in vvox.app, and then there's a meeting number that's also in the chat, you can copy and paste in. You'll be able to answer the questions there, and we'll provide the results at the end of the talk.
Okay, I'll move on. So then if we look at roundworm fecal testing in practice and what they do in other countries and how could we implement it here in Ireland, there seems to be two trains of thought if you look at the literature and key opinion leaders. And one of it would be a replacement for treating, or the second way we can use it is testing alongside treatment. And why would we do that? So that can help, reduce parasiticide use, which combined with that risk assessment, which is, overtly important, negative tests can be interpreted to not treat the animal at all. And then the opposite of that is that when you get a positive test, it's a positive reinforcement for the client to actually treat an animal that is high risk, or if the animal is positive and they're living in a household that has a high risk population, that you actually can say, here are the eggs, you need to treat this animal. So it can also reduce environmental contamination associated with the use of parasiticides. So in large animals, there's definitely a lot of research in regards to, the residual activity of these parasiticides that we use in the dung of sheep and cattle and horses, and the effect it has on dung beetles, and the effect it has on grass growth, and the effect it has on the micro-ecological system around that dung pat. And so we have to assume that that is, similar or the same in dog feces. We're not 100% certain, but, you'd have to assume that it does have an effect on the ecosystem around. And then also when we're testing, we're getting local information within our practice and national information and surveillance. And with global warming and pets moving around more, that testing gives us an idea of what's coming in and out of the area and may alert us to, foreign parasites that are not usually found in Ireland. And then this is really important, and I think it's going to come to the forefront more and more in Ireland and England. So like I said, like Niamh said, I'm in Australia at the moment at a conference, and hookworm is a massive problem in Australia and America, especially in greyhound populations. And resistant hookworm, can cause severe disease in dogs, but also it lives in the soil and you can get infections through your feet. And the only real way to know that your lung or hookworm treatment is working is by doing fecal egg reduction testing. And the World Parasitology Organization is going to release guidelines to say when you're treating for hookworm, you should really be doing a quantitative fecal reduction test. And then obviously doing that testing assesses compliance. So it makes sure that your wormer is working, and it's a positive reinforcement. And I think that's undervalued, that time that you spend talking to an owner about their dog or their cat, and when you're asking them about their risk assessment, you often might pick up on something else. And plus, I think during COVID there was a paper shown that was published that the effect of seeing a worm in the poo of a companion animal has a massive effect on that animal-client bond, and a very negative effect. And then lastly, as Jason had alluded to, there is the potential of zoonotic disease, and so we need to be very mindful of that. So the limitations then to roundworm fecal testing in general in practice is, the first of all is getting the sample. And there is a paper that showed that, or survey that some owners will actually go out in the middle of the night to let their dog poo on the street rather than in the daytime, so when they have to pick it up. The next thing then is that how often does it need to be done? And again, that becomes part of your risk assessment. You do need a test that's highly sensitive. And if you're doing it in-house, then obviously you need the equipment and you need training and the time that's necessary to do it. And then the expense to clients. So, these tests cost money, and oftentimes they cost more money than the wormer themselves. So that's a big barrier. And then that changing of the culture within the veterinary practice themselves and then within clients. So just say you do have the poo, what do you do next? So when you collect it, then refrigerate it. And then I think really after, preparing for this talk and talking to people, I think you really have two options. You can do it yourself in-house or you send it away. And I just took the pictures. I was in the Philippines recently launching the oversight, and these are cats and they live outdoors. And this is like a really expensive designer shop, and there was Louis Vuitton, and they just live there and people feed them, but they're really big on fecal testing. But I'm just wondering where these cats are pooing, which is quite scary. But they're massive over there on fecal testing, and they're aware of the zoonotic potential in children, and children get wormed quite regularly in the Philippines. So let's say then you want to do a microscopic fecal examination. You can do that in-house, and there's a number of ways of doing it. So you can just do a direct fecal smear, or you can do a fecal flotation just passively, or you can do fecal centrifugation and flotation. And the flotation fluid that you use is super, super important. So it has to have a specific gravity that allows the eggs to float to the top. And what has been shown, if you look at this paper here, is that if you look at the difference between a direct smear and the OVA assay, so the OVA assay is like kind of a test that comes with its own zinc sulfate, but you don't centrifuge it. But you can see the massive difference between not centrifuging and centrifuging. And then there's a slight difference between zinc sulfate and or zinc sulfate flotation and sugar flotation. So people do this a lot, definitely in America and Brazil and the Philippines. They will routinely perform fecal flotations in the practice. But, it does come with, you need training. And obviously there's some people that are experts at it, and then other people, that are doing it that probably with the practice get better and better. But it does take a certain amount of input from the practice, creating standard operating procedures, a dedicated prep area, because it can be quite, dirty and messy. And obviously then trying to keep positive controls to ensure you get training and comparison would be useful. So the limitations of a microscopic examination or just doing a standard fecal egg examination would be the false positive. So dogs love to eat poo, and sometimes you'll see your ruminant strongyles through feces, or sometimes Eimeria, and sometimes some toxicar eggs, and that will go through like a marble. So they're not actually infecting the dog or the cat, they've just been eaten and they're just passing through. Or misidentification, so the heart-shaped structure at the bottom of the screen is actually a piece of pollen. And then you can get false negatives. So obviously, only adult female worms produce the eggs. So if you've got a pre-patent infection, then you'll have a negative sample. Intermittent or low egg shedding, again, depending on the sensitivity of your fecal flotation, you could miss them. And then the sampling itself, how good was the sample? You know, obviously they say you should really take 3 samples over 3 days. And then back to that operator, you know, the practice, the experience, and the exposure to looking at these eggs. So then you have the coproantigen testing. And this is available through IDEX. And this is where you're testing for adult worms, and it's adult worms and larvae, sorry, and it's an antigen. So, you know, it's a very specific and very sensitive test, and it will give you a diagnosis in that pre-patent period. And also it avoids that false negative result with intermittent shedding of eggs, and it rules out those dogs and cats that are, you know, eating poo of sheep or other dogs and cats. The problem is that it's expensive and you do have to send away the sample. But CAPC, which would be the American Companion Animal Parasitology Advisement Board, they would recommend really as a gold standard that you combine the two, the antigen testing and the fecal testing. And if you look at the results of the coproantigen testing versus the fecal egg testing, it did get 10% more samples that fecal egg testing missed. But overall, the agreement with them was similar, and in less than 1%, the treatment outcome differed. So whether you were going to treat or not. But actually, what I found the most interesting about the recent study that they published was that out of 898,000 samples, I think 83% were negative. So, you know, it's quite rare to get a positive test, and that's what we found in the company that I work for when we're screening and testing is oftentimes, you know, you're getting negative samples. So then, how the heck do I move to testing in my practice? So for me, a big thing would be familiarizing yourself with the risk assessment. And I have a slide, the next slide, on the VetSustain UK risk assessment template. So it's getting to look at the client and the family that the dog and the cat is in and the lifestyle. And then whether you implement that into a routine health check and a sick patient check. So it can be part of both. I think if you're going to try and implement fecal egg testing and use that risk assessment in your consult or out at the front desk when you're dispensing parasiticides, it's really that training in-house and getting everybody familiar with asking the questions. And as you get used to it, then it becomes much quicker. And then I think, you know, it's going to be client-led. So definitely here in Australia, I was in a clinic with a good friend of mine today, and you know, she was saying that clients are moving away from using parasiticides and putting things into their dogs and cats, chemicals, and they want to move away from that. So that is the VetSustain, the two risk assessment tools that are available on their website, and it's really interesting. And then I'll just briefly mention the oversight pet. So I don't know, some people might be familiar with the oversight as a ruminant and horse fecal analyzer, but now we've launched the oversight pet, and it does in-house fecal testing for small animals as well. So these are my dogs and one cat. So thank you so much for listening. And then we have one last question, and I'll leave that up for a second. And if people could take the time to fill it in.
Well, Sarah, first, if you could just let me share my screen and I'll show the results of the last one.
Oh, sorry. Spoiler alert.
Share this, and the results have been counted and verified, and they are in. And most people, 61%, rarely or never perform fecal egg count testing, which is what I probably expected the answer to be. About 7% do it for all, and 15% only when clinical signs are present, and then occasionally. Oh, can you see this? Yeah. Grand. So they're the results, which makes sense. So I'll go on to the next poll. Next poll. Oh, we'll just do that. Hang on. So if you want to read this after today's webinar, what change, if any, might you consider in your approach to parasite control in dogs and cats? So you can just pop a short phrase there, and the results will come up. So feel free to enter those in your at your leisure. And I suppose we can go to some chat and questions now.
Yeah, amazing. Thank you very much, Sarah and Jason, for that as well. It was a really, really great talk from all three people there. So shall we go to some of the questions? There has been quite a little bit of activity anyway in the Q&A. So let me see. I think the first one seems to be from Jeff, and it's a question at the end about the current stance of DEFRA/VMD in tackling the levels of fipronil and imidacloprid in rivers as a result of pet treatments. Suppose that one might be for you, Rose.
Yeah. So they recently published a roadmap. So this was the VMD, the Veterinary Medicines Directorate, which is aiming to tackle the pollution. I think it has sort of short, medium, and long-term components. It is a very, very cautious approach. So I think their short-term strategy is to consult with stakeholders and I think to raise awareness around the pollution. And I'm not sure exactly what the concrete plans are to do that. In the medium term, I think they were planning to sort of gather more evidence. I'm not sure what the evidence that they're planning to gather. And in the long term, I think there was discussion around regulatory change, but that's very much a long-term plan. I think basically they agree that the EMA should review the regulatory framework. But one thing that's just worth mentioning is that even if the regulatory framework is updated and the phase 1 and phase 2 risk assessments are sort of strengthened, this would not be a retrospective thing. So it would only apply to new products. It wouldn't really affect the currently licensed products and the sort of pollution that we're seeing from fipronil and imidacloprid.
Yeah, and that's the concern, isn't it? I'm just actually looking at some of the results coming in there, Jason. I'm enjoying the buy a goldfish, whoever put that in.
Probably the most environmentally friendly pet you could get.
Probably, by the looks of it. Rose, the next question was actually from myself, and it's when you were mentioning kind of contextualized care and that it's a requirement now for prescribing parasiticides in the UK by the RCVS. I was just wondering, have you heard how this change has been received by practices and owners in the UK when this was brought in? I was working as a vet over in the UK when this was actually coming in, but only for the first couple of months. And I remember there was a lot of upset around it between owners giving out and also kind of vets, you know, an increase in workload for vets and everything. But I'm just wondering, has that settled down? What's the current thoughts on it now?
Yeah, I think, to be honest, I think change has been pretty slow to sort of filter down to the practice level. To some extent, it's because the guidelines are quite vague. So it's very, there are many interpretations of the word blanket treatment. Their main guideline is that we should be avoiding blanket treatment, which to me means that we shouldn't be routinely treating all animals. But I think what's often happened on the ground is that people, well, I know a lot of vets have switched from spot-ons to oral flea and tick treatments, or sort of do some sort of assessment in terms of what treatment they should be given, but they're not moving away from just treating everything. So everything is in many cases, all pets are still being treated, but maybe with slightly different regimes. So it's a slow process. I think there's a lot of uncertainty and quite a lot of defensive practice going on, which is completely understandable. I still see practice, and I know part of the problem as well is that pet owners are not necessarily aware of the changes, so they expect to be given the and worm kill ectoparasite and endoparasite treatment. And it's quite a long, involved conversation to explain to them why we might be changing that. So it's not easy for vets in practice to change. I know some of the more progressive practices are bringing these changes on board, but I think I wouldn't say that's widespread yet.
Yeah, and I think my experience was that there was a big concern about the loss of revenue. From it, and I think when it was brought in, the main thing that we were told when we were over there was just to get the wording right in terms of the prescription, so that we'd be able to blanket cover them for 12 months once they were seen at their vaccination appointment. So yeah, it's slow progress, I suppose. But The next question was from yourself, Rose. Do we know the percentage of Toxocara species eggs in parks that is coming from foxes? And we have a response there from Amanda. The Toxocara is surprisingly low in the foxes we've tested in UCG. Capillaria, hookworm, and lungworm were in almost 50% we tested, which is actually quite interesting. Do you know anything about that, Jason?
So when we were doing it, we genetically tested every single egg we found to see if it was either cat-eye or canis. But obviously you can't tell the difference between the the canis that comes from a dog or the canis that comes from a fox. So it's the same species. So we couldn't tell. But the modelling studies that Eric Morgan has done suggest that foxes would be responsible for a very, very tiny amount of the environmental contamination with Toxocara eggs. It's mostly pet dogs that are likely to be responsible. So they're the ones we should concentrate our, control strategies on.
Yeah, brilliant. Just having a look here. If anyone else has any more questions, right, and feel free to add them in. We have from Hilda, are the oral forms of parasiticides like Bravecto or Simparica better as regards less environmental residues? That's actually an interesting one. Probably for yourself, Rose?
Yeah, that's a question I get all the time. And the most honest answer is we just don't know. And because they have no environmental risk assessment, I think they probably are less likely to have an impact, although it depends on a lot of factors. So the advantage of the oral ones is that they're mostly excreted in the poo. So as long as you're picking up and disposing of your dog's poo, which you should be doing anyway, then hopefully most of it is going in the bin. But I do have concerns around them. So they are extreme. The isoxazoline, so the newer ectoparasiticides, are extremely persistent in the environment. So they're actually much more persistent than fipronil and imidacloprid. So they're actually PFAS chemicals, so per and polyfluoroalkyl substances known as forever chemicals. So they hang around for a really long time. And there is some evidence that they might be more potent. So they also behave differently. So they're much less likely to go into waterways, but because they're more lipophilic, they're probably going likely to bind to soil particles. So that there's definitely a risk of soil contamination and soil accumulation. But yeah, the bottom line is we just don't really know where they're ending up.
And it would be great if we could get retrospective data on it, wouldn't it?
It would.
Yeah, I don't think we'll have the powers to enforce that though. You did mention there as well, Rose, that there is a critique of the evidence for routine or blind worming of adult pets for Toxocara in Vet Sustain's FAQs, which is available online. I know Sarah mentioned some of the risk assessment tools, but they have a lot of resources on their website, including this frequently asked questions. And the one part that I found quite interesting, I never realised, was that some studies have found that routine worming has no significant effect on the shedding of Toxocara from adult household dogs and cats, which, yeah, I just never realised that. Do you have any comments on it?
Yeah, it's not something I realised until relatively recently. It was actually Martin Whitehead who gave a talk on this at BVA Congress, and he's gone through a lot of these studies and found that, yeah, there are several studies that looked at the effect of routine worming on Toxocara shedding, and actually found there was no significant difference between dogs that were regularly wormed and dogs that weren't wormed. There are a few studies that I think found associations, but most of the effective worming, I think, is worming that's done in younger animals, so juvenile animals. So if you look at the Vet Sustain resources, you can see that we've advised worming of juvenile dogs, so blind worming of juvenile dogs, I think, is quite evidence-based, but it's a lot more questionable, I think, to be routine worming adult pets. But in certain circumstances, if they're in kennels or shelters or perhaps a bit immunocompromised, it would make sense. But I think, as I say, pretty questionable to be routinely worming adult dogs and cats, as far as I can gather from the evidence.
No, I probably agree now. Like I don't know about the studies that he was talking about, and maybe it could be something to do with that most dogs are zero, and some dogs, and depends how routinely as well. Like if it's just 4 times a month, then there is always a window for the worms to creep in. So if you were doing it 12 times a month, I would be surprised if. So not 12 times, but 12 times a year, I'd be surprised if there was no difference between.
Oh, well, so some of those studies did include monthly worming, and they didn't find an effect. So
They didn't find it. So like they were still So that would imply that they had resistant.
Well, I think one of them found a slight effect, but it wasn't statistically significant. And the other thing that they found was that actually a really low prevalence of toxocara. So the most recent one was it's called the dogpox study from 2022, and they looked at toxocara occurrence in dog feces samples from around the UK, and they looked at various parks like Manchester and all around the UK. And I think they had 304 dog samples and dog poo samples, and they found no toxocara in any of them, which is pretty interesting. It does make you question why we why we are routinely treating animals when we're not really.
I found one positive sample in 46, which is okay, higher than 0 in 300, but we did find eggs there, and I think that there beside the entrance does imply it's the pet dogs and not foxes, you know, responsible for most of this. Now, maybe not.
But yeah, that was why I asked the question about foxes, because we know foxes do shed toxocara, and if it's just not occurring in dog feces, you know, it may well be coming from foxes. I think Eric Morgan's study, which did modelling on that, found that if people were picking up and disposing of their dog's poo, then fox poo would be the main source of egg shedding in the environment.
But then was it not the sheer numbers of dogs, like the volume of pet dog poo would be considerably higher than the volume of fox poo going into the environment? I suppose there's lots of foxes now.
Yeah, but once again,
It's
if people are picking up dog poo, then fox poo becomes the next bigger.
Are they picking it up? It doesn't look like it.
Maybe not in Ireland anyway. In the UK, maybe it's better.
Oh, it's everywhere. They complain about it everywhere.
So Kaylee has asked, how often per year would you recommend testing pet feces? Sarah, do you want to chip on that one, or?
Yeah, so I suppose CAPC would say 1 to 2 times a year, depending on the risk, and ESCAP, I think 4 times a year. But it just goes back to that risk assessment. It's all about the pet. So, you know, it's looking at the pet and what the pet is doing and where the pet's going, and then you go from there. In my opinion, you know, it's not like there's a set rule. So, yeah.
I mean, like, to have perfect information, you'd want to like test it every month.
You would,
you know, like,
yeah,
and then you'd be
would.
doing it 3 consecutive days, 12 times a year. Nobody's going to do that, like. But you know, if we want good data, that's what we have to do. But any data is better than some of the data. We don't have that much data at the moment about what happens in pet dogs. So more testing will give us more data.
Exactly. And I guess the actual likelihood of owners bringing in their dog's poo once a month is, in my experience, low, but tell me if I'm wrong. We just have a comment from Amanda there saying local authorities should be made aware of their dog breeding establishments in their county where infected puppies are being sourced by owners. We're seeing too many puppies testing positive for multiple parasites with zero accountability from DBEs. Dog breeders they are being sourced from, GRD being the top of the list. I completely agree. Like, we're seeing a huge amount of GRD and different other things being found in puppies, like just lots of roundworms when they come in. What are your thoughts on any of that? I agree with it, but I don't know how we'd go about doing it.
I mean, it's even a problem with the rehoming. That's how the resistant hookworm got out. You know, they think in America, where the greyhounds came from these kennels, where, you know, they're living almost like horses and cattle and being wormed. And then they, you know, they weren't tested, and then they went out into the general population. Yeah, I'm not sure. I think the dog walk study showed that Giardia was the top of the list as well. So that's not surprising. I don't know, maybe when you're doing your puppy packs, that it might be an idea to start to look at whether or not you include that as a test. You know, I don't know. I know it's putting the onus on the owner.
And Paul says it's not the local authorities that are responsible for that staff. Yeah, I guess that is probably a bit that's probably above our pay grade for answering, knowing where to go with that one. I think yeah, it is a tricky one with the amount that we are seeing coming in puppies and everything. It's definitely food for thought anyway. And then one final question there, just from Jill. I think it was the first question that we had, just about the VMD approach and why is their approach so cautious given the evidence now available of levels above PNECs in the majority of rivers. Rose, do you want to take that one as well?
I think you'd have to ask the VMD.
Yeah.
I couldn't possibly comment.
And then, is there any way to reduce environmental shedding after treatment of dogs? Oh yeah. Do they, if they're increasing shedding? I suppose just picking up their.
Yeah, there are some things that people can do. So, you know, don't allow your pet to swim if you buy a spot on, I think is a pretty good rule of thumb. I mean, people have suggested things like having covers on their beds and throwing it in the bin instead of washing it, but I don't know how practical that is. But it is important to follow the label instructions. So things like don't stroke your dog in the sort of 24 hours after applying a spot on, to wait until it dries. That'll reduce the emissions. But yeah, there's only so much we can do to reduce. A lot of the emissions after applying spot-ons are pretty unavoidable, unfortunately. And the best thing really is just not to use a treatment that isn't necessary. And I think in quite a lot of cases, it's arguable whether routine treatment is necessary.
Brilliant. And I think that's all the questions there. So thank you very much. I'll just pop up my final 2 slides. Will be really quick because I know we are after running a little bit over time. So just bear with me for 1 second there, everybody. I'll just present. Yeah, can everyone see that? Yeah, all good. So just to finish off, a big thank you to everyone for attending, and our speakers for giving their valuable time to us this evening, and again to the committee and Excel Vets who helped organise this event. I hope this has shown you how important it is to consider the environmental impact of the decisions that we are making every day as veterinary professionals. Just a brief word on the EMA public consultation. So the submission deadline is the 31st of October. We at the Veterinary Sustainability Network are going to make a submission just in light of their proposed methodology for developing guidelines in terms of companion animal parasiticide legislation. And we want to make sure that our voice is heard. I'd encourage any other organisations throughout the industry to do the same, just to make sure that we are actually getting our voice out there. Finally, we'll send out the resources mentioned in this webinar tonight via email over the coming days, and a recording will be available at some stage on our YouTube channel in due course, which we'll send out the link to as well. CVE or CPD certificates will follow pending VCI approval, so hopefully we get that. And then just very finally, just before we finish up, I'd just like to let you know how to get involved if you do wish to do so. If you'd like to stay up to date with our activities, please sign up to our mailing list using the QR code on the right. We also have our profile on Instagram and on LinkedIn as well, and then our website is up and running as well. And then finally, as we are a not-for-profit, we do rely on the donations of others to keep the show on the road at the moment. For anyone that would like to donate, please find the donation link via the QR code on the left-hand side. And yeah, with that, I shall let you go. Thank you everybody for their time this evening. I really enjoyed tonight's session, and I hope this has inspired you to look a little bit differently at our kind of way of prescribing parasiticides for companion animals. So yeah, Thanks a mil. Cheers.